Okay, thank you. So moving on to our second topic for today from the laboratory, it's my very great pleasure to introduce Dr. Carolina Pavlovsky from Argentina, who has already made her presence very much felt. Carolina is a very well-known physician in the region. She is the head of the research department in FUNDALEU, Buenos Aires. Welcome, Carolina.
Hi.
You're quite present already. And our fourth and final expert today is another giant in the CML field, Professor Jerry Radich, who again has very much made his presence feel throughout the TFR session. Thank you. He heads up the Clinical Research Division and is a Kurt Einstein Endowed Chair at the Fred Hutchinson Cancer Center in Seattle, USA. Welcome, Jerry. Such a privilege to have you here.
Sure.
So Carolina, if you would please share your slides with us. And as you're getting your slides up, I'll let the audience know that we'll run this session a little bit differently to the last one. So Carolina will spend a few minutes identifying key issues, problems, and unmet laboratory needs in Argentina. And then she and Jerry will pretty much take turns highlighting some of the key issues or unmet laboratory needs and how they can be addressed. We will be open to questions and comments from the audience throughout the session. So again, please use the virtual hand or the chat function and let your voice be heard. Thank you, Carolina.
Okay, thank you, Arlene. Thank you, Nicola, and the CML Foundation Education Program for this invitation that I always like to be here with you and collaborate. And as you ask to me and to Jerry, this part will be mostly not a talk, but commenting our issues, challenges, and unmet needs in the region. And as you know, with the widespread availability of good quality TKI in the developed world, the survival of patients with CML in chronic phase is approaching to that of the general population. The survival is the same also. However, the situation in our countries, low in some middle-income countries, may not be so easy. Why? No? Because not only the problem is the TKI reaching an ideal TKI treatment, but also the problem with the monitoring of this disease. That is very important. Mostly, I think this is the most important thing to talk about. And well, some issues of our region. The first one I thought was a diagnosis. What happens at diagnosis? Patients, when they are asked PCR, perhaps the PCR is qualitative, the qualitative reverse transcription PCR, or quantitative. And sometimes doctors ask for what they have, for what they can, because they cannot afford perhaps a quantitative PCR. But we know that the qualitative PCR is recommended at diagnosis to perform and detect the type of transcript, and the quantitative also, it's a good tool at diagnosis, and also a critical tool for disease burden. In our institution, we use the quantitative PCR, and this we know that provides an accurate measurement of the leukemia cell mass and the degree of the protein P210, BCA-ABL transcript, and we can know from where we start, and then we compare in serial monitorings, and we know how the curve is going down. So, but this has perhaps pros and cons. So if this is negative, we must look for other isoforms. What is the issue in our region? Sometimes and mostly, not mostly, but I would say that I have realized this when we started the protocol of discontinuation in Argentina, where one of the criteria was to have the PCR or qualitative or quantitative diagnosis. And sometimes patients couldn't enter the study because they didn't have one of these two molecular studies, because the patients that were diagnosed a long time ago, perhaps they were diagnosed with a cytogenetics and they weren't, this was not performed. So now it's very important to make it now in the TFR era. And the other thing is the thing that's not affordable, perhaps, for public centers. Another important thing that I would like to talk also with Jerry and discuss is the importance of the cytogenetic diagnosis and during follow-up. This was a paper published a long time ago, and there are a lot of papers about the abnormalities in the cytogenetic clones. But this curve shows the abnormalities in the chromosomes of the major root, and you can see in the top curve in the first group, the trisomy 8 and the extra phi, for example, that have an overall survival better than those of the other group. And this was mostly when the alteration, the clonal alteration was a single clonal alteration. So this is an important point because sometimes we do not perform cytogenetics anymore. If the lab we have is standardized and the qPCR is available, perhaps the cytogenetic analysis is less essential for response assessment. But clonal evolution, however, rarely occurs without increasing transcript concentrations and probably inducing a loss of hematologic response. So sometimes you say, if you have a good PCR, if you have a PCR that you can trust and is reliable, must we make cytogenetics every 3 years, every 2 years, or as the guideline says, if the patient has no dysplastic symptoms or anemia or plaquetopenia or any cytopenia? Really, this is interesting to know. Also, we can say that in our region, it's better to make a cytogenetic analysis that not make anything, because perhaps we don't have access to PCR. And we can make a cytogenetic. In my institution, I have access to PCR, to quantitative PCR, but this is not the reality of all the region in Latin America and Argentina. So affording treatment is not the only problem when economic conditions are not ideal, and monitoring is an essential tool. And let's talk about the molecular monitoring to measure the transcript in international scale. This is very important. And the importance of the serial monitoring to have every 3 months a PCR can let us observe the kinetics of the molecular response and also the late responders or those patients who have a rise and how it and the behavior of the transcript. So I think it's very important is the serial monitoring analysis. And the standardization also, it's an important method that every lab must be standardized because it reproduces all the results in the same way thanks to the conversion factor. So issues in Latin America, molecular analysis is not available to all clinicians and still many labs cannot access to standardization. So this many times impacts on the treatment decisions because we are making a decision, perhaps when a patient comes with a loss of MMR of different labs, or perhaps a high transcript level, for example, 10% or 6%. And when you measure it in a standardized lab, it gives you 1.6 or 0.5. So there are a lot of variabilities when the labs is not the same and it's not standardized. So I think one of the important things to take into account is to make efforts to standardize labs as well as centralize. This is one objective. And the use of different units of measurements and laboratory protocols makes it difficult to compare the results of different labs. The possibility of inter-laboratory variation continues to be an issue. This is a very important thing, and I don't know, Brazil and Argentina, I know how it's standardized, but the rest of Latin America, I really don't, I'm not sure if there are many standardized labs. This is a little bit about what Katia told and spoke very well in the emerging regions outside clinical trials. In our trial in Argentina, we performed this trial because of this problem. When we started in FUNDALEU thinking about this with Beatriz Moiraghi, Ana Varela, and Michele Bianchini, that are the 4 persons who wrote this trial,
we start thinking it's better to make a trial in Argentina to take care about our patients and guarantees make them have the PCR monthly. So that's what we do. But what happens about outside clinical trials? And outside clinical trials, there's not a proper centralized monitoring. The access to serial PCR and the rapid turnaround of the results are not perhaps made in the best way in public hospitals. And also the patient perhaps is not much compliant when it's out a clinical trial. Sorry, this is outside. And another important, just to finish, another important test that I also want to know to talk with Jerry is the mutational analysis, because in Argentina we have very few labs that are making it. We have a program free that thanks to that program, the patients can make the mutational analysis. Now we have stopped that program and we're going to start again. But the patients on public hospital don't know where to do it. So what happens? Selects the best TKI adapted to the mutation detected. It's like something that must be done. We must have, because if that patient has a T315I mutation and you still keep giving a second generation TKI, the patient will have a real failure and we must change to ponatinib. So this is a very important point in the region. Also, the challenge is to have access, to this mutation analysis and the unmet need in the region, and that what we were trying to perform, and unfortunately we don't have perhaps the funds the money to do it, is the next generation sequencing and digital PCR. That these are one of the things I would like also to discuss with Jerry. So some regions have a high variation in terms of economic development, health profiles among its population, and geographical barriers. Argentina is a very long country for those who know it. From north to south. So the challenges are the lack of resources, the pure distribution. Most patients have the possibility to make a lot of things of this in the capital, but not all around the country. And the variability of the health system, private versus public. And also we have a lot of copies in Argentina, some of them not of very good quality. So with this, I finished my first part, and I don't know if you would like, Arlene, I can continue sharing and we can talk with Jerry, or how would you like to continue?
Jerry, would you like to chime in on?
I could pipe in on a few things. Sure. That was great. Thanks very much. I'll just a few things. So the first thing is that even though obviously I'm extremely interested in the molecular biology and testing and monitoring and the like, the one key thing is that what we've learned from things like the GPAP experience, where patients are given TKIs but aren't necessarily monitored very well, is their survival is great. So the key thing is getting people drug and getting them compliant with drug. Once you do that, that has to be, if you're going to design any health system, that's the main thing. Get them drug, make them take their drug, right? One of the things that molecular monitoring does, and you can argue whether or not Q3 months is best, Q6 months is best, et cetera. One of the things that I think that molecular monitoring does the best and why it improves outcome is our insistence to the patients that they get monitored carefully, I think tells them on a deep level that taking their drug and looking at their response to the drug is hugely important. So I think if nothing else, the emphasis in molecular monitoring emphasizes to them that the importance of taking their drug consistently. And like, most good students, they don't want to fail their teacher. So if they know that we're watching by monitoring, they'll do their homework and then take their drugs, right? Then the issue becomes, what happens when you have suboptimal things? There are a lot of places, it sounds like not as much around in Argentina and South America, but in Africa, for instance, it's often easier in a lot of places for them to get a PCR test than to get cytogenetics. Because they can do peripheral blood PCR and find a Cepheid machine, but finding a bone marrow biopsy needle and a lab that can do cytogenetics is really, really hard. So there are some people in limit countries where you have to monitor their PCR and their peripheral blood and really limit cytogenetics in those patients whose disease is not behaving as you'd expect. So, for instance, if they get a PCR of the BCR at diagnosis, and at 3 months, if it isn't falling at all and the like, then those are people that you might say, okay, this is the person that we have to invest in doing the cytogenetics to, the people who aren't acting normally. There are patients where you can't get a PCR, but you can get FISH. And in that case, I think one thing that's helpful to remember is that most of the survival benefit in all of the studies is at the 1% level. That's where you get most of your survival benefit. If you go deeper into MR, 0.1 and below, that has some improvement, but the lion's share of the improvement in survival is at the 1% or MR 2 level. You can get that with FISH if you convince people to do enough FISH, right? So most FISH, at least in this country, are done on 500 cells. If you bribe your FISH person to do 2 plates of 1,000 cells, you get to basically a 1% to 0.1% sensitivity. So you can get to pretty good sensitivities if you just convince the people to do more interfaces or metaphases on FISH panel if you don't have a PCR available. Other issues that were brought up was the importance of the international scale.
Yes. The importance and perhaps I would like to ask you more about the clonal evolution and the alterations in the because this is sometimes like confusing when this appears at diagnosis or during the treatment. No.
Yeah. It's very different, right? So at there's, as you know, there are many different definitions of accelerated phase. And the WHO is trying to change that. Yeah. In most of the CML chronic phase studies that were done, the IRIS trial and the like, they actually allowed people with cytogenetic abnormalities besides affiliated positive to be counted in those studies as chronic phase. So they didn't count those against those. And I think that there's enough small studies out there that show that at diagnosis, a lot of the patients with cytogenetic abnormalities do quite well and behave similar to chronic phase. I would, myself, when I see those, I'm a little bit more suspicious and follow those people even closer than I would a normal cytogenetics patients. And typically, if someone with diagnosis shows up with other clonal abnormalities, I start an unrelated donor search to find a donor in case we need to go to transplant early. But it's certainly different than people who get cytogenetic changes while on therapy. Those people, that's tantamount to going into blast crisis. Those people do terribly. And those are patients who, as soon as you see a clonal evolution on therapy, should first probably change tyrosine kinase, but certainly should be headed towards a transplant. Because the chance of curing those patients who evolve into new cytogenetic changes while on therapy, those are really bad patients. If that answers that question.
Excellent. I will change the slide. Yes. Yeah. Do you see this? Can you see it?
Yes.
I want today. I had a case. A person, a doctor that made a consult about a case that I have put there in the slide. that she was talking to me and telling me that it was a resistant patient, no? To imatinib, nilotinib, and it says andasatinib, and now it's indasatinib. And the patient didn't have a molecular response. I think it was, the patient had 20% of BCR ABL. So she made the mutational analysis and appeared an deletion of exon 7 in the gene ABL1. And I was thinking about this because these are the questions I start thinking with our group of molecular and cytogenetic in FUNDALEU, because as I told you, we analyzed 9 patients with these variants and the rare mutations, and there is not much published. No, we are preparing the manuscript, but can you comment something about this?
Yeah, it's been seen a variety of times. There's a lot of labs that don't even report it when they find it because they don't know what to do with it. I think its clinical significance in this case is suggested just by the patient's history. I mean, and I think someone who's resistant to imatinib, nilotinib, and dasatinib, they may respond to ponatinib, but I think that the chance that they're going to get a long-term response to ponatinib is extremely slim. And I would think this would certainly be a transplant candidate. This person is I would bet EHAB that this person is not going to do well. I know EHAB also has lots of money to lose, so I'm willing to take his money for him.
And you would win. And I think just on the ponatinib, in this case, I would definitely plan for transplant, and I would, based on the optic data, would do ponatinib 45 to start with, and then reduce to 15 when they respond. But I would start out with ponatinib 45.
You would change to ponatinib?
Yeah, I would change to ponatinib and ponatinib 45, not 30, and then plan for transplant.
Yes, and the other, there is Rocio, I think, Rocio de la Mer, who works in my institution and in the lab. She wants to make a comment. Perhaps Rocio, you want to ask something?
Yes, I would like to ask, in these cases where we find variants that they are not well studied, or we don't really know the sensitivity, it would be correct to do molecular docking, or do you use any in silico tools to kind of try to find an answer of which would be the best treatment?
Yeah, we have not done anything so that sophisticated yet at all. Currently, our work on all of these are more technical and really don't have any clinical applications yet. We're doing a lot of data with the so-called high-seq sequencing, where we try to find the molecular partners of cytogenetic changes. And so finding that people who look to have simple cytogenetic changes actually have extremely complicated cytogenetic changes that you just can't pick up on cytogenetics, and trying to use that as a way to understand. And that might actually be true. We think even in these people who look like they just have the Philadelphia chromosome, but they just do terribly. And the question is, do those people actually have a lot more going on cytogenetically than the chromosome? The extreme value of that is that the group I'm working with did one of these analyses on someone with breast cancer who had a simple inversion of one chromosome. And when they actually looked hard enough, they actually had almost 100 different cytogenetic lesions that were buried in the chromosome that were just too small to pick up. So it was like complete genetic, even though it looked simple. And I suspect that once we start using these fine things, a lot of these cases that look like they should be behaving normally, but don't, I think there's going to be a reason for it. I think that when you start looking structurally at the genome, it's going to be very different. And Illumina is coming out with things now where I think, getting to some of your point of cytogenetics and the like, I think probably within the next 5 years, we'll be able to do whole genome next generation sequencing fairly cheaply and be able to get not only point mutations and the like, but also translocations and structural differences. So I think probably within the next 5 years, fish cytogenetics are going to be out, that we're going to be able to do one assay and get really point mutation all the way to structural stuff in one assay. I think that's really I think that's going to happen. I'm usually pretty doubtful, but I think that's actually going to happen.
And affordable for all.
I think eventually it will be. Yeah. I think that there's enough pressure being put on a lot of these places now, from efforts like the International CML Foundation and all the people that are out there. I think that they're realizing that it can't be an exclusive club anymore. That's just not going to wash.
Yes, we need to work together. And from my opinion now, because I think we're in time, no? But if anyone wants to make a question, si alguien quiere hacer alguna pregunta antes puede levantar la mano. Just a comment for you, for all of you. I think it's important to work together from our region to United States, and the CML Foundation, it's an important link. But also we have the LALNet, Latin American Leukemia Net group of all our Latin expert. Katia is part of it, and Jorge Cortés is the president. So for me, it would be very important to make something that perhaps permit us to make a research project with what you're mentioning.
Yeah, I mean, we would love to help. We're spinning out a new machine that we think is going to be cheap, that we're sending to Uganda, which we think it's going to be very cheap digital PCR, and it's based on using old CD recorder motors, which are really cheap nowadays. You can get that for a couple of bucks. But you can spin samples by using a CD thing into thousands of separate digital PCR wells, and then do isothermal PCR. It looks pretty nifty. So there's going to be solutions. I think we just have to. I just think where there's a will, there's a way, and I think that if we all work together, there's enough people out there, even at these companies, who are sympathetic. I think we just have to find them.
Bruna.
Thank you, Jerry. There's Bruna, no? Bruna wants to ask.
There's a paper of Simona Soverini from 2020 about including the NGS panel early when the patient is on warning or a failure. Instead of performing the Sanger sequencing, you should perform NGS, and to detect small amounts of cells with mutations that would not be saw in the sequencing, traditional sequencing. Yep. Sanger, and hypothesizing that you should incorporate the NGS instead of sequencing to detect these mutations of low levels earlier, and then you could switch the TKI earlier or adjust the dose and offer the patient a better approach. What do you think about incorporating this approach?
So there is a lot of debate about that. And at the International CML meeting in France, there will probably be a formal debate about that. Because, it's very attractive. There's not a lot of data. I mean, there's certainly not a lot of data to enforce that. One of the questions that's going to come up when you really think about it is when you're talking about sensitivities of, how deep are you going to look for some of these mutations, depending on your technology. Next generation sequencing can pick up like a 5% clone. That probably is real. Some of the more sensitive techniques can pick up 1 in 10,000. Most of us out there right now will have many of these mutations at a level of 1 in 10,000 already. When we've done, just so-called chip mutations, when we've looked at using duplex sequencing, which can find these myeloid mutations in 1 in a million. CHIP is in everyone, even young kids. You will find it everywhere you look, pretty much. So what you're going to have to do is really look at the structure of your disease resistance and the structure of your VAFs of your mutations, right? So if you have a BCR-ABL that's in the 5% level and your AXL1 is down in 1 in a million, it's probably not driving that resistance, right? Yeah. So it's going to be, and then you also have to realize that VAFs are not accurate from gene to gene. So when you look at a VAF of an NRAS is 40% and a VAF of a FLT3 is 20%, you actually can't accurately say that those are two different clones. In fact, the wobble on measuring each of those clones is each of those targets is quite different. And so when you start to put all that together, there's a lot of noise in the system as it exists right now. And I think you can make some inferences, but I don't know if we're ready to drive therapy based on it quite yet. I think it's one of those things where I'm doing it on newly diagnosed patients, but not for just to get to the knowledge. I'm not using it for anything right now. If you were to diagnose someone who looked like they might be towards high risk to accelerated phase and you found that they had an AXL1 mutation, would I do anything different? No, but I'd probably watch them more carefully and I might change the bar in when I would think about changing therapies, right? But I would probably be more suspicious in how I watch them. But I don't think I would necessarily commit them to a transplant route or anything else. But I would just, put that as one more piece of data interest in following people. That's a long-winded answer, sorry.
I think that, Jerry, that perhaps NGS, it would be useful in the moment, perhaps when you have suboptimal patients. This is a day-to-day problem that we say those patients who don't achieve MMR, for example, but they are not a failure. So we all want to know if there is a clone there, a malignant clone, but perhaps by NGS you can catch that and say, well, I'm going to change it to asciminib or and start thinking of a transplant. For that patients, I think it would be perhaps interesting in the future. No,
I think what it's going to be useful is, we have, let's say at least 50% of patients who have some resistance have no mutations in ABL kinase, right? So those people always do badly, right? Compared to people who have a tyrosine kinase resistance. If you have a point mutation, you can always switch to another drug. If you have a resistance when there's no evidence of ABL mutation, they're presumably having resistance by some other biological mechanism that changing a TKI isn't necessarily going to help. And so if you do mutation stuff, you might be able to see another mutation that would make you worried. You may not have a drug that would help unless they had an IDH mutation or something. But I would think it might help you be more suspicious that this is someone who's going to need more dire therapy, like a transplant or something down the line.
Okay.
Bruna, do you have a follow-up or is it an old hand?
That's the last.
It was an old hand. Lillian has put a question out. Lillian, do you want to talk to your question?
It's a clinical question about TFR because always exclusion criteria is resistance to any TKI, first line specifically. And after switching to patients resistant to imatinib, we have patients with deep molecular response for a long time, many years, and that's not included like a criteria for starting TFR. What do you think about that?
So yes, it has been an exclusion criteria in a lot of studies. You're correct, except we do have from the eNest stop study that did include both resistant and intolerant. So a small number of patients, but if you look at success of TFR in those groups, in that specific studies, there was really no difference. So I would feel comfortable stopping those patients and monitoring them the same, which is the monthly and then every 2 months for 18 months and then every 3 months. So I would feel comfortable in someone who's achieved a good response.
And somewhat similar to that, in a patient with an adverse event, a serious adverse event like a vascular events, cerebral ischemic attack, do you use that as a criteria to start earlier the TFR, or don't you?
You mean someone who fits the criteria or doesn't fit the criteria of TFR?
That does, or had a resistance, and basically the resistance.
Yeah, so the same thing. If they're resistant and then they responded to your next one and had achieved a deep enough response, I would stop them.
Can I ask you another question about a clinical case, a patient with a TFR that has been without therapy for years and is starting to rise the PCR and has a MR2, and when I repeat it, it has MR3. And then 2 times MR3 and then MR2, and that goes down. Do you restart therapy in those cases?
So I think your question relates more like, are late relapses happen? And yes, late relapses do happen. And there was a recent paper on late relapses, even 5 years out, even 10 years out. And that's why the continuous monitoring is really important.
But
yes, I do. Go ahead.
If the patient spontaneously lowers her PCR, you restart?
It looks like they're between 0.1 and 1. I would restart with the caveat of like what Jerry was saying earlier. Can the patients live like this for a really long time? Yes, it's possible. We just don't know the answer to that. Remember the natural history of CML. I am actually old enough to remember that. The natural history of CML chronic phase, when it's detected, is 5 years. So can someone live with that 0.1 to 1 for a really long time? Yes, possible. Would I recommend that they restart? Yes, I would.
Yes.
Okay, we might have one last question from Bruna and then some concluding comments perhaps from Carolina and Jerry.
It's my last question.
You're on fire, Bruna.
About, I know it's probably, you would say that it's a old discussion and probably dead discussion, but about the BCR ABL with the insertion of 35 pair of bases as being a product of an alternative splicing. I know that the most discussions about that say that it's not a problem. It's a function that the product, but do you think that it could confuse the monitoring because we can see in the PCR a small amount of cells harboring these BCR ABL with insertion, and it could confuse. For example, you should think that that patient is failing or raising the log, and actually he's not. He's still can continue on TFR, etc. What do you think about that?
This has been a perplexing issue for ages. It's still unclear to me. I last talked to Sue about it, Branford, just a month ago. And she says she doesn't even report it out anymore. She doesn't want it. She thinks it's not a deal, and closes her eyes, closes her ears to it. I always worry about it. I haven't figured out a good way to study it yet. But until then, I'm not changing behavior of what I tell patients about it.
Yeah.
If you think of any good ideas, well, let me know. I don't have any.
Carolina and Jerry, do you have some final comments?
Just say that we need to continue monitoring. We have to select, PCR is like the most important thing to monitor our patients. Make great efforts for the standardization. It would be interesting to see in LATAM which are the ones who are standardized. And this is the first step to the last one, that is TFR. So it's all linked from diagnosis, P to 10, TFR at the last. So I think this is a very interesting conclusion. And the second point of my conclusion is what we have talked about, what Jerry said, and these important things about the mutations and the variants and the deletions. These are important things perhaps to publish or to discuss in this kind of meetings, because we don't know what to do or if change, not change. So thank you very much for EHAB, Jerry and Katia also, because it was a nice meeting.
No, I think thank you everyone for the good deeds you're doing. I'm resolute and hopeful that the work that we're doing, the work that the CML Foundation is doing, we've got a lot of work to do to kind of bring state-of-the-art care to the rest of the world, but I think we're doing it, and I think we can do it. I think I'm a bone marrow transplanter. I have to be optimistic by nature, right? Otherwise you wouldn't do this stuff, right? So, but I think that we can get it done. With the will is a way.
That's great. Thank you so much, everyone. Wow, that last hour and a half just really disappeared. So thank you so much, Carolina, Jerry, Katia, and EHAB too, for such a lively discussion. And thanks also to audience members. I do have a couple of questions here that came in by email that we haven't got to. I will get you a response and get back to you. And likewise, if you have some questions or comments after the fact, please just email them through to me. I will get some responses for you. So we hope to see you soon. There is one more scheduled Latin American discussion on Thursday, the 25th of May, where the topic will be the wide-ranging challenging scenarios, and another great team of experts there leading those discussions, as you can see. I will email everyone soon with a link to register for that discussion, and also pointing you towards the Knowledge Center if you're not already familiar with it. There is literally a wealth of knowledge in there. There are dedicated modules on TFR from the laboratory, as well as CML therapy, management practicalities. There is the first tranche of a pediatric CML module, the second half to come soon. We have a module on managing accelerated and blast phase CML, which is almost ready to go, and a pregnancy module not too far down the track. Oh, and I'll send you the link to Climb for a Cure too, and enjoy. So I think with that, I will say thank you, gracias y obrigado to everyone. Thank you so much.